which is biological molecules. Now, there is a class of molecules that are known as organic molecules. And these are the ones that contain primarily carbon and hydrogen. These two elements, and also sometimes oxygen and nitrogen. So these are the key players. Of course, the main is basically carbon along with hydrogen. And the reason they're called organic molecules is, one reason is because they are found in living organisms. Excuse me. So they're found in living organisms, and there are a variety of them. Right? And what we're basically concerned with is perhaps the class of these compounds, but that's more of a topic for chemistry. You've got the alkanes and the alkenes and the alcohols and the aldehydes and all of that. So we're not going to go into those details, of course, right? Chemistry is where you'll be dealing with those. Right, so we also know, why are we studying this? Because we need nutrition. And the substances that we eat, that we consider food, right, the food substances are primarily either carbohydrates, proteins, or lipids. Right? And of course, as you could guess, all three of these are examples of organic molecules. Now, there are different types of carbohydrates and different types of proteins and different types of lipids. Right? So we'll mention a bit about that very briefly, but right, we're just going to introduce that. Okay, fine. These are the three classes of compounds that are considered, like they're present in the food we consume, and the food that is overall mostly consumed like in nature, right? So any organism, even if it's not the regular kind of organism that you would think of. For example, bacteria, right? So they also go for, right, these organic molecules. And then there are nucleic acids, right, which are the molecules that make up the genetic material of an organism in the cell, right, which is basically the DNA and the RNA. So these are the four that we'll be talking about. And then, right, food tests. So how to identify whether a sample that's given to you contains either of these. Because nucleic acids aren't the food. Nutrition part is for the carbohydrates, proteins, and lipids. So identifying carbohydrates, proteins, lipids. And let's start obviously with carbohydrates. These are made up of carbon, hydrogen, oxygen. There are many different types, ones that are made up of three carbons, so three carbons, five carbons, six carbons, right? The ones that are made up of three carbons are called trios. The ones with the five are called the pentos, and as you could guess, the ones that are made up of six carbon atoms, are called hexols. So, right, a simple representation of a hexose is like this. Right, this is a simple representation of a six carbon carbohydrate, for example, glucose. And the formula for glucose is C6H12O6. Right, for pentose, you can represent it like that. Right, so that O is an oxygen. Right, so this guy is an oxygen. So this is what the molecule looks like. Very simplified, of course. I'm not pointing out everything else that's going on over there. And an example or two examples that are relevant for pentose are ribose, which are present in RNA molecules, and deoxyribose, which are present in DNA molecules. In fact, the D in DNA is deoxyribose. The R in RNA is ribose. Right? Now, these guys, right, are known as monosaccharides. Mono meaning one, and saccharide meaning sugar. Right? So these are single sugar molecules. What happens is, right, these guys can build up into more complex molecules. So you can have two of them joining together. In what is called a condensation reaction. And that's basically meaning that when two molecules join together, there is a loss of a water molecule. And then they form disaccharides. Right? Di meaning two, saccharide meaning sugar. So these guys have two molecules. And so the sugar molecules that, right, the monosaccharides that we will be concerned with are glucose, as already mentioned, galactose, and fructose. Right, and two of the disaccharides that we'll be concerned with are maltose. In fact, I'll write down three: We're concerned with lactose as well, and sucrose. So maltose is a sugar, right? It's made up of two glucose molecules. It's the sugar that's found in bread, for example. Lactose is made up of glucose and galactose. And this is found in milk. And sucrose is made up of glucose and fructose. And this is the sugar that's found in plants, and also the white or processed sugar that we consume, that is sucrose. Any questions? Okay. Now, as you saw, the fact that a single, like two molecules can join up to form a disaccharide, similarly, right, multiple or many, right, up to thousands of monosaccharides can join to form what are called polysaccharides. Poly meaning many. Okay. So we're not putting a number on this because they can be hundreds, it could be thousands. We're not saying, oh, these many make up that molecule. Also, to keep in mind, when we talk about sugar, like this is a carbohydrate, right? Polysaccharides are carbohydrates, monosaccharides are carbohydrates, disaccharides are carbohydrates, right? So when you talk about sugar, we are basically talking about mono and disaccharides, right? Polysaccharides are not called sugars. Okay. The reason being, and this is not mentioned in your book, but still because sugars are sweet and they are soluble in water. These are two, right, reasons, whereas polysaccharides are not sweet and they're not soluble. Right, so if you want to, you can show it up like this, that this circle represents carbohydrates and these guys over here are sugars. Right, so you can say all sugars are carbohydrates, but all carbohydrates are not sugars. Does that make sense? Okay. The other thing, right? Yeah, go ahead. They are not. Well, well, a potato is sweet. It's not like you shouldn't compare the sweetness to the sweetness of that white processed sugar that you consume, because that is like concentrated, right? And plus, it's a disaccharide. Disaccharides and monosaccharides are sweet. Achha, I say potatoes are sweet. They contain the polysaccharide called starch, which we'll just talk about. Why is it sweet? Because as soon as you put food in your mouth, digestion begins. And so enzymes start breaking up the molecules in the food, right? In the mouth, it's basically carbohydrates. So as soon as those start being broken down from the polysaccharide to the disaccharide, you will start feeling, that sweet taste in your mouth, even though if it's like, so if you take a piece of potato, or you take a piece of bread which also has starch, and you keep it in your mouth for a minute or so, you just like roll it around, it will start tasting a bit sweet. If you're eating bread, there's a slight sweetness when you're chewing it. Not saying it's a lot, but there is. Okay? Right. Now, we've seen that these guys can join up, and right, the polysaccharide is an example of a polymer. Right? Poly means many, and this means unit. So polymers are made of many repeating units or molecules of monomers. Right? Now, if you're catching the term mono meaning one and bi meaning for single units, join up to make molecules that have many units. Right? And so polymers can be huge. Right? They can be very, very big. Now, as far as carbohydrates are concerned, the polymers that are concerned with the polysaccharides are cellulose, starch, and glycogen. Right? These two guys are found in plants, and this is found in animals. So let's talk about cellulose. Right? It is a polymer of glucose. In fact, all of these guys are polymers of glucose. Write that over there. So it's a polymer of glucose made up of thousands of monomers. Right? It forms long, straight chains, and it has high tensile strength, which means that can withstand a lot of pulling or pushing. Right? It's not going to break easily. It is strong and rigid. And we can understand these properties because, right, we just talked about how the cell wall does not allow the plant cell to expand when water is entering, and it protects the plant cell sort of. It gives it structural support, right? And so they're present in cell walls, not only of plants, but they're present in cell walls, and they provide structural support. Then you've got starch, right? This is the storage form of uh, carbohydrate. As I already mentioned, it's a polymer of glucose. It is a storage form of carbohydrate in plants. So you would find it in individual cells, and you will also find them in storage organs like the potato you mentioned yesterday. Potatoes are super rich in starch, right? Rice is rich in starch, right? Wheat is rich in starch. And it's also the storage form of carbohydrate in seeds, right? Because seeds need nutrition when they begin to grow, when they germinate. So starch is present. It is a branch molecule, right? So it's something like this, and right, it sort of purrs up, right, to become compact for storage. And then we've got glycogen, and its properties as far as your O level is concerned are similar to starch. It is the storage form of carbohydrate in animals. Right? It is primarily stored in the liver. It is branched, in fact, highly branched, and also like curls up to be compact. Right, and so glycogen is what comes into play when your body needs sugar and you haven't eaten for a while, and so this is converted back into glucose to keep your blood sugar level normal so that you keep going. After a meal, obviously you consume more than you need, and so the extra sugar is converted to glycogen. There is a limit to that because anything over what the body can store as glycogen is then converted into fats, and that's how we gain weight. If I just right, so they're present in the cells and storage organ, obviously, and seeds. Any questions? Okay, so after that, all good. We move on to proteins. Well, actually, lipids first, I guess. Lipids are mentioned first in the text. Now, we don't really go into detail. Lipids are made up of molecules that are called fatty acids. They contain the elements carbon and hydrogen, oxygen. It's basically carbon, hydrogen. The oxygen comes like at the end. There's just two of those in the entire fatty acid molecule. It's all carbon and hydrogen. And lipids are made by when fatty acid molecules combine via condensation reaction. So what I mentioned for carbohydrates. For each condensation reaction, you have loss of one water molecule. So they combine with a molecule that's called glycerol. Right now, there are different combinations that can occur, but what we are concerned with is what's called a triglyceride, and I'll tell you why that's called. So a glycerol molecule has three carbons, right? It has hydrogens, right? So H2, right? It's not, this isn't the correct way of putting it, right? It's got hydrogens here and here. This guy's got one hydrogen and then one here and then one down here, which you could barely see, right? And then it's actually an OH. So this is glycerol. This is what glycerol looks like, right? But after the reaction occurs, the H of the OH is gone. So I'll erase that. Right, the chemistry part of this is not in your syllabus. and then you have fatty acid molecules at each of these positions. So you've got three of them. Okay, and this guy is called a triglyceride. So tri meaning three, and glyceride meaning glycerol molecule. So this is a glycerol molecule with three fatty acid chains attached to it. It doesn't always have to be that way, but that's the one that we're going to talk about. Now lipids, if the lipid is a liquid at room temperature, and we're talking about like standard room temperature, so 25 degrees, it's called an oil. And these guys mostly come from plant sources. And if it's a solid, it's called fat, which mostly comes from animal sources. Now, lipids are very important because they make up the membranes in the cells. So, lipids make up the cell membrane and all the membranes on inside the cell. So, the membrane that is surrounding the mitochondria, the membrane that's surrounding the chloroplast, the nucleus, other membranes that you don't know, right? Golgi apparatus and endoplasmic reticulum. So you don't, like, I just mentioned that you don't really need to know those, right? And so internal membranes of cells. So for example, the mitochondria, probably that's an organelle in the cell, the nucleus, the chloroplast. Any questions? Okay. And sometimes there are lipid droplets that are also used as an energy source in cells. All right, next up we have proteins. And these guys are polymers made up of amino acid monomers. So the monomers are called amino acids, the polymers are called proteins. There's another name which sort of suits the mono and right. So proteins are sometimes called polypeptides, and the amino acids are called monopeptides. That's just something you can know. Now, proteins play a very important part in an organism because they are present pretty much everywhere, right? So enzymes, all enzymes are proteins, a lot of hormones are proteins. You've got proteins in the cell membrane, right? Other signaling molecules. and organelles, so the ribosome or the mitochondria, you can, I've given the book. Right, and so they mention a bit about enzymes, which is the next chapter, so we'll do that next time. And right, they control chemical reaction in the cells, so we'll talk about that. Now, the structure of amino acids isn't relevant. They are made up of 4 to 5 elements. It's again primarily carbon and hydrogen, but there's also oxygen, nitrogen, and sometimes sulfur. There are 20 naturally occurring amino acids, right? And they join up via condensation in specific sequence to form the different proteins. So in amino acids, in proteins, the sequence of the amino acids is very important. You can have, right, for example, 1, 2, 3, 4, that's 1. You can have 1, 3, 2, 4, 1, 3, 4, 2. Even though it's the same 4 amino acids, but the arrangement, the sequence will all have an effect. So if this is a protein, then all 3 of them are different. You can't say, oh, it's the same amino acids, it's the same protein. It doesn't work that. Any questions? Okay, so they can be small chains, they can, be hundreds to thousands of monopeptides long. Okay, lastly, as far as biological molecules are concerned, there's nucleic acids. And basically, in nucleic acids, they're talking about DNA. All right, so not talking about RNA. And it's made up of the elements carbon, hydrogen, the sugar that's involved. So there's a carbohydrate that's involved. In RNA, it's the 5-carbon ribose. In DNA, there is the 5-carbon deoxyribose. So you've got carbon, hydrogen, oxygen, nitrogen, and phosphorus. And right, the monomer is called a nucleotide. Right, so DNA molecules are polynucleotides. And as far as DNA is concerned, it's made up of 2 strands. Has 2 strands and forms what's called a double helix. So its shape is sort of like a spiral, right? So that's one strand, and that's the other strand. It's not obviously very well made, but something like that. And right, the nucleotide is made up of a 5-carbon sugar, which I mentioned, right? A phosphate group, which looks like this, PO3, and what's called a nitrogen base. It's made of phosphorus. Do you see the P over there? Right? The phosphate group is made using the element phosphorus. Any questions? Okay. From here, let's quickly go through food tests. So we're done talking about biological molecules. There are obviously many others, but these were the ones that we are concerned with. So first up is testing for the presence of starch. If you give in a sample and they're going to ask, oh, does this sample have starch in it? How are you going to check that? So you take that sample and you mix it with warm water, right, in a test tube. In a test tube, you have sample plus warm water. So obviously not using like large quantities of these things, and you mix it. And once that's done, it will form a suspension, and then you add something called an iodine solution. So it will form a suspension, and then you add about 3 to 4 drops of iodine solution. If starch is present, right, you will get a blue, black, oops, not black, black, right, coloration. And that's it. Then you've got the Benedict's test. Now, something to know about this test. This is a test for reducing sugar. Right? What are reducing sugars? Reducing sugars are ones that act as reducing agents. So little chemistry over there. Right? So they become, right, the sugars become oxidized, and the Benedict solution is a copper 2 solution. And that is reduced to copper 1. Okay? So this is a test for reducing sugars. And you see a change in color from blue to red. So copper 1 is blue, copper 2 is blue, copper 1 is red. The thing is that for the purpose of our syllabus, all mono and disaccharides are reducing sugars, except starch. Sorry, starch the polyol. Except for sucrose. Right? Sucrose is non-reducing. So what they're going to ask you to check whether sucrose is present or not, right? And also all polysaccharides are non-reducing. So if they're saying, oh, is sucrose present or not, you're going to do this test. If it does form a red color, that means it's not sucrose because sucrose does not do the reducing thing. Or if they simply ask you whether it's a reducing sugar or not. If it does give you the red precipitate, that means it's a reducing sugar, so it's not sucrose, it's not a polysaccharide. If it remains blue, right, that means it's not a reducing sugar, right? So that means either a sucrose over there or it's a polysaccharide, or perhaps it's not even a carbohydrate, or maybe it's like a negative test. Anyway, what do? You take a small volume of, right, the sugar. In this case, we're taking glucose so that we know the reaction is happening. So we take glucose and we add an equal volume of Benedict solution. And we heat it up, we warm it, right? So you want this thing to be warm. This is the only test which requires heating or warming, okay? And when you do that, you will notice a color change. It will go from blue to, as they mentioned, cloudy green, to yellow, and finally to brick red, right? So red. I already mentioned why that's happening. The copper 2 is being reduced to copper 1. So because glucose is a reducing sugar, it's a monosaccharide, is a reducing sugar, you will see this happen. Is this clear? Okay. The third test is testing for proteins, and that is called the Biuret's test, right? For proteins. And so what you do is you take equal volumes of your protein sample and you mix in again equal volume of dilute sodium hydroxide and an equal volume of copper sulfate solution. If there is uh, protein present, you will notice that the color will change from blue to purple. To go from blue to purple. And that means protein is present. And lastly, you have the test for lipid, which is also called the emulsion test. And what you want to do over here is you want to take a sample of whatever lipid you have, and you want to add ethanol, that's an alcohol, and so the lipid will dissolve. With this, right, then you add this to a few centimeter cubes, so a small volume of water. If lipid was present in your sample, then a milky white emulsion will form. Right? So if a lipid was present, it will give you a milky white emulsion. And then you okay, fine. What is an emulsion? Right? An emulsion, it's a mixture of two liquids that don't normally mix. Right? So I'll just write that down over here. Of two liquids that don't normally mix. And obviously the prime example is water and oil. So you can try this at home. You can take a little bit of oil and you can add it to water and you can shake it. And so what you will notice, right, is if you've taken more water and a bit of oil, so you've got water over here, and normally if you add water to oil, it forms like a layer on the surface. Have you ever seen that? Right? But if you mix it, then you will see like oil droplets scattered. So they're not dissolving, they're not mixing, but they're scattered in the water. That's an emulsion. It's a mixture of two liquids that don't normally, so they're still not mixing. So the oil becomes scattered in the water. If you leave it for long enough, it will come back together, right? But that's what an emulsion looks like. So you can try that out with a little bit of oil and water at home as well, and you can see how that turns out. In this case, it will, right, because you mix the lipid with the ethanol, and so you're getting like a milky white color, right? Instead of adding it directly to the water, right? And there's like more chemistry behind it, which is again right beyond the scope of the syllabus, so I won't be going into that. Any questions? Then that is the end of the chapter and end of class, and I'll see you next time, inshallah. All right. Allah.